University of Chicago.
All right, I want to thank the organizers for giving me a chance to present my work. The title of my talk is Primate Transcript and Protein Expression Levels Evolve Under Compensatory Selection Pressures. So I thought I'd start out with the broad motivation of this work, which is really in the context of evolutionary genetics. It's to contribute to our understanding of phenotypic differences between species, as well as contribute to our understanding of human-specific traits. So comparative studies of gene regulation, both in primate cell lines and tissues, have identified many transcriptional regulatory differences between primates. And this has really been driven by advances in high-throughput sequencing and related technologies, which allow you to assay binding of different factors, open chromatin regions, DNA methylation. And they've really these technologies, as well as these studies, have placed comparative studies of gene regulation at the forefront of research in evolutionary genetics, especially in the context of studying phenotypic variation both within and between species. Yet, ultimately, proteins perform the work of the cell. Their levels are very likely the direct targets of selection, and they're more coupled to phenotype. And there are a number of known post-translational regulatory mechanisms that are independent of transcriptional regulation. For instance, post-translational modifications such as phosphorylation and ubiquitination. And here, the dominant technology is not high-throughput sequencing anymore, but high-resolution mass spectrometry. So just to highlight some previous work in this area, 2 studies have shown across diverse taxa that protein levels are more correlated across species than absolute protein levels are more correlated across species than our mRNA levels. And this prior work has some limitations. The absolute estimates are often not accurate enough to identify small, statistically significant differences between species in individual genes. There are technical issues with the array platforms that were compared. And it's not entirely clear what this means for closely related species, for instance, primates. So we conducted a study in which we looked at lymphoblastoid cell lines derived from 5 genetically distinct individuals from human, chimpanzee, and rhesus. We analyzed the samples by high-resolution mass spectrometry, as well as RNA-seq. And we analyzed the resulting data in the context of orthologous exonic regions, which were filtered for duplicated regions, paralogous regions across the species. So just some more detail on the protein quantification. What we did was we used stable isotope labeling in cell culture, SILAC, and we labeled up a human SILAC cell line and used that as an internal standard to compare protein levels across the individuals in the species. So the sequence divergence is sufficiently close that we can get individual peptide quantifications overlapping the same sequence, from which we derived median level relative protein measurements across the individuals and species. So we also use a similar strategy for mRNA quantification. We RNA-seq the SILAC internal standard, as well as each individual, and computed log2 RPKM ratios relative to the standard across these orthologous regions, and then normalized and derived mRNA level measurements. So just to describe one of the limitations of mass spec, so here on the left I'm showing the number of proteins quantified in 3 individuals across all the species, or for different numbers of individuals. And in our study we analyzed 3,390 genes which were quantified in 3 or more individuals in all 3 species. And shown on the right are the distribution of all the RPKM values across the species, and then in green are those quantified in 1 or more individual at the protein level, and then in purple are proteins quantified in 3 or more individuals across all 3 species. So the mass spec data is really only sampling highly abundant or medium to high abundance proteins. So that's a limitation of current mass spec technology. So the first thing we did was we performed differential expression analysis in which we used the replication in our study to identify genes that are significantly different, both at the mRNA and the protein level. So this is just an illustration for 3 individuals. And one of the sort of striking result was that we found more differentially expressed mRNAs across species than proteins at an FDR. So this can be explained by a couple possibilities. I'll focus on point one here, potentially that the protein levels are just less accurate and precise. So we chose SILAC on purpose here because SILAC has been shown to provide essentially a linear response across a large range, and the response you get matches the ratio you expect. So here is a peptide quantified by biochemical means and then spiked into a complex background. And the response matches the ratio you expect. So some of you may have heard of technologies like iTrack and TMT, and these quantify proteins in a slightly different way and are subject to ratio compression. So the plot you see on the left there will be angled. So we chose SILAC to make sure the results are accurate. Another thing that we did to evaluate the sort of precision of the measurements is that we looked at the ratio you get from distinct exons from the same gene. So averaging over a splicing signal, you expect these ratios to be correlated between human and chimpanzee. So if anything, our results suggest that the protein data are more precise than the mRNA, or no worse. So there are sort of two issues that remain. They're either higher within species variance at the protein level or a slower rate of divergence in protein levels between species. So to address this, what we did was we used essentially our replicate measurements of human-chimpanzee divergence, which we can get by normalizing out the standard for both mass spec as well as RNA-seq. And now we can ask if these divergence estimates differ significantly across the technologies. So when we do this, we find that among genes where mRNA divergence differs significantly from protein divergence, there's an axis of genes where mRNA divergence is higher than protein divergence. So we did a number of analyses to make sure this result is robust. It's in the manuscript. But from this sort of key point, we argue that protein expression levels might evolve under greater evolutionary constraint than mRNA levels. So next, using our rhesus macaque samples as an outgroup, we looked for signatures of selection on gene regulation. So there's sort of two broad approaches to doing this. The first approach assumes a null model and then tests for deviations from this null. So this is limited in that you can't estimate in non-model organisms, primates in particular, parameters of that model like mutation accumulation rate. So there's a second approach, which is more heuristic in nature, in which we look for cross-species expression patterns that match a certain evolutionary scenario. And it only provides a list of candidate genes for further study. So we initially looked for expression patterns that are consistent with stabilizing selection. So these are patterns that have low intra and low inter-individual variation. And then we rank these on variation from from low variation to high variation. And those are the top of the list, the top 200. You can choose any cutoff, are candidates for further study. So what we find is that when you look at the sort of top-ranking candidates using mRNA measurements, that consistent with prior studies, these candidates are coupled with measures of regulatory constraint. So here on the left is the NDS, and on the right is the number of protein-protein interactions. Yet when we look at protein levels ranked on these intra and inter-individual variation, the effect is much more pronounced. And if you look at other measures which are correlated with these measures of regulatory constraint, we see the same sort of bigger effect. So we can also look for lineage-specific patterns in our data. So on the left here is DDX24, which is a human-specific increase. And then on the right, we have a chimpanzee-specific decrease. And interestingly, the human-specific protein levels are enriched for genes or proteins involved in transport and localization, which are distinctly post-translational processes. So we also have both RNA, we have RNA-seq as well as mRNA data, and we can also look in our data for more complex candidates, more complex evolutionary scenarios, genes that fit more complex evolutionary scenarios. So on the left, you'll see the raw data, so the RNA-seq read coverages, and on the bottom are individual peptides that were measured across this gene. And on the right are the per individual measurements. So here we have an example of a gene, a candidate, for a human-specific change or human-specific increase that could be attributed to a change in post-transcriptional or post-translational regulation. Yet the predominant pattern in our data are cases where we have a lineage-specific change at the mRNA level, yet very little variation across many peptide measurements across the gene. And we argue that this pattern is consistent with compensatory evolution. And just to sort of wrap everything together, our results reveal that the proteome is likely under greater evolutionary constraint. Thus, many of the changes you see at the mRNA level are effectively neutral if they're compensated for at the protein level. And we are also, for the first time, using this joint data set, able to identify regulatory signatures that are consistent with compensatory selection. And really the next step forward is to use the genomics technologies that we have at our disposal to understand the mechanisms by which these patterns actually arise. And with that, I'd like to acknowledge my postdoctorate supervisors, Yoav Gilad and Jonathan Pritchard, as well as the really great people who I worked with, and some funding, and then Michael Ford, who provided incredible collaboration as well as amazing mass spec services. And if you have any questions and ideas for collaborations, I'm starting a new group at Maryland. Please do send me an email. Thank you.
Thank you for a very, very nice presentation. So, I mean, there's been a lot of evidence out there, or at least a lot of suggestive evidence, even if it's not conclusive, about this whole turnover and buffering of individual mutations and so on. But I'm just wondering if the only evidence in your particular case was the discordance between the mRNA divergence and then the protein divergence. I'm just wondering if there's any other evidence, for example, if you looked at translational sites or sequence regulatory elements that might suggest that indeed there is a difference in translation efficiency, or if there were differences in, codon usage or you name it. So something that is more than simply, oh, perhaps there's more noise in the mRNA measurement level. Because, I mean, I do agree with your conclusion. I'm just trying to see just how...
So we've tried a couple other lines of evidence. So one, we see more evidence of relaxation of constraint at the mRNA level, so where the variance increases. So our study is also highly replicated, so we're not really, it's not really noise, we're measuring actual differential expression. So I think that the previous studies that look at just correlation are seeing the hint. Our study kind of really provides really much stronger evidence using replication that this phenomena exists.
And your idea is that it's at the translation level, or at the degradation level, or at the transport, or
so we don't have mechanisms in this case. We just have these expression patterns and candidates for genes. So ultimately, I think that the genes at the top of our list are candidates for functional study. So I think the challenge at this point is to really figure out what mechanisms drive this.
And I'm just wondering, I mean, you guys have also looked a lot at, regulatory elements, like non-transcriptional. I'm just wondering if you're finding a similar story where there's more divergence at the, regulatory regions than at the mRNA level, which would suggest, again, this sort of buffering at all stages.
Say yes.
So it's more complicated, but I'll definitely talk offline about that.
Okay, so you said you in general found that there was often a lot of divergence in mRNA and then less divergence in your mass spec. I was wondering if you found exceptions to that where there was as much or more divergence in the protein data, and if so, was there any pattern as to what those genes do?
So in the so I actually showed one example where you see reduced expression in the human lineage just at the protein level. Mm-hmm. So we found some weak, I mean, we're really stretching the protein data here, but we do find some weak correlations with a number of phosphorylation. These genes tend to have more phosphorylation sites, ubiquitination sites. So hints that these are more, maybe they're more regulated at the protein level. So we do see it, but it's really pushing the numbers at this point.
Great, thank you.
Thanks.